microscope.
How many
Smile, take your pictures, no yelling.
Okay, so welcome everyone to this tissue culture review session. I know you guys had a lot of questions about this. So first of all, I just want to go over real quick the terminology and everything. I also give you guys some note cards. So if you have any questions, please put it down. And then I'm gonna hold like a few 2-minute breaks where you can ask your questions. So if you need clarification on something, just let me know, or if it's really urgent, stop me. I'm more than happy to answer it. Okay. See if this is my clicker is gonna work. Okay, so basically, why would we do tissue culture? Basically, you can get some high volume plants, basically monsteras, something like this. You can propagate a bunch of plants with relative ease. For example, I'm doing some research, and from one plant I have to regenerate like 60 or 70. Instead of taking me 2 years to do that, I can use tissue culture and regenerate that 70 plants in 2 months. So that is a huge reduction in time and square footage that would require me to achieve that. We have some also other high value plants like orchids. Usually these are award-winning orchids, and that would reduce its value if you cross it. So it has to be asexually propagated. You can take your plants to like a flower show, and the judge is gonna evaluate it. And if you win, your plant is basically super expensive. And most of these are also like charity auction. That's why they are so expensive. So basically, what are we trying to do?
Can you go back up real quick?
Yes.
$5 million rose.
Basically, these are charity auction plants. So, you know, like rich people, it's a tax write-off for them basically, but it goes to charity, right?
Oh.
They bid over something that someone donates, and basically what you pay for it goes to a charity. So it's not like an actual $5 million rose or not a $220,000 plant. It's just basically some rich guy buys it and writes it off as a tax because it's
And then they get the plant.
Yeah. Basically, it's mostly that. But if you want that exact plant, you can't really cross it. You can't do anything else. You have to use tissue culture to propagate it. So what we're gonna talk about today is what is tissue culture, or plant cell / tissue culture. That's gonna be like a really big thing. The definition, the micropropagation, what we do with it. We're gonna talk about a little bit of GMO and the virtual filter. That's basically the lab tour and stuff like that. We could do that online too. Okay, so the tissue culture techniques, micropropagation is what we call it when we regenerate one plant or multiple plants from one plant cells. Basically, if you're gonna get virus-free plants, you wanna get disease-free plants, you have some issue going on that you can get rid of with normal propagation methods. You can get the apical meristem, the top of the apical meristem, and you remove that. And from those few cells, few undifferentiated cells, you can grow callus, which I actually got you guys some callus, this white stuff. This is my corn research, so please don't open it. But I mean, we have plenty of lines because from like a size of a dust, like I do embryo rescue or embryo isolation. It's technically not rescue because that embryo rescue is a little bit different. But basically I get, yes.
Keep going.
Okay, so basically what I do when the corn, after pollination, after 12 to 14 days, depending on weather, once the kernels get big enough, I sterilize the kernel, cut the seeds open, and get the immature embryo out. Put it on that media, and then I'm gonna get callus. Yes?
So, embryo. Plants?
Plant. It's a plant.
What is the embryo?
So you know how you have the seed. Okay, let me.
I'm
So
sorry.
you can.
I can.
You can use both the apical meristem
And-
and or an.
Yes. So let's see.
Embryo.
That's not gonna get it.
You know how you have the seed, which
Okay,
has
This
the.
is the seed, right? This is the corn.
Let's hope it's not gonna.
Oh my god.
So
okay, yeah,
basically, when you have the embryo, let's see. You have the.
There's more to it than just.
Okay. So the embryo would be. This is not that good
So
picture.
imagine like.
Okay. So basically, this is the.
There you go.
This is a bigger picture, but this is your food storage. Basically, this is what it grows. And the embryo, this tiny thing where you have the pollen and the ovary, where the ovule,
So.
like the seed, like it's the actual, not the storage of, not.
So you're taking out the like, what part is?
So I'm taking out this tiny part.
Okay.
Usually this is a lot smaller than the actual. What we use mostly from the seed is the storage where the embryo grows from. The embryo uses all that energy storage to push out roots, push out shoot system before it can photosynthesize. I'm getting the embryo, and if I think I can form embryo.
I know, I think I understand that. I was wondering, we talked about embryos a lot, so for a lot of micropropagation, it just.
Yes. So basically, this is the whole, like this is the whole corn seed, right? And when I get it, I want to get it before it reaches this big size. When it's like this tiny thing, before the seed actually gets mature. Because when it's mature, it's hard and dry. And when it's like a size of a dust particle, basically, when it's that small, you scoop it out. You don't need the energy storage anymore because it's gonna get all of the nutrients and sucrose, which is the energy when we created the media. We put it in, it's gonna use that. And from that, we just gonna undifferentiate it, and you're gonna have the callus.
And what kind of tools do you use to harvest your immature?
A little like, I can show you guys next week, but we have these little tiny scoops. And if you're really interested, I can buy some sweet corn and I can show it to you guys.
So there was a word that he used a lot, I think it was spontaneous embryogenesis or something like that.
Yeah.
Is that
so-
meaning that they're creating, not necessarily using the embryo, but making, is that any of the culture that made new roots, or was that specific to actually using the embryo? And then maybe I have to go find the word again and then have my paper, so.
We definitely interrupted the flow of your thing. I just.
Okay, so embryogenesis is when like you basically abort them, when you do a cross between two species that pollinate, but they can produce a viable offspring.
Okay.
So that's when you need to do an embryo rescue, because you will get the seed, but the embryo is not viable without tissue culture. And like I said, we can do virus-free plants, we can do germplasm preservation, which is like, we basically gonna use like either magenta boxes or these little tiny tubes, and you're gonna have huge populations. Like this would be an individual species. It could be a tree, it could be a corn plant, it could be anything. And instead of taking up so many square feet, this is it. Takes up this much space, like you guys saw during our tour. And you can do haploid and double haploid plants for actual breeding purposes.
You said after you take the apical meristem or the immature embryo, the cells then differentiate and become the callus?
Yes. So when you basically remove it, from the seed or very when you remove the embryo or whatever cell it is, callus is a response for bonding, right? When you cut it, it's like a response to avoid any kind of infection. That's how it heals itself. And when we equal auxin and cytokinin, then you're gonna encourage callus growth. You wanna make sure to remember auxin and cytokinin and how these ratios, because if you have more auxin than cytokinin, you're gonna encourage root growth. When you have more cytokinin than auxin, then you're gonna encourage shoot growth. When those are in equal amounts, then you're gonna encourage callus formation.
On that, you already make the culture, as you make the agar or whatever,
Yeah.
the medium, you already put in your
Yes.
hormones. So do you move the plant at time? Because let's say you put in the equal to get the callus.
Yeah.
and everything. Do you move it from there after you have callus like you have
Yes.
there? And then you put
Yes,
it
you
in?
move it to a different plate.
Okay.
That's how you can manipulate it.
Okay.
It's really easy. You open it up, you just scoop it up and put it somewhere else, which is a different kind of media.
And also, to go back to what you were saying before, you were going with, you could either take the apical meristem or the embryo to do
Yes.
these cultures. Can't you, you can take any part of the plant to do this.
Yes, that's correct. You can do any part of the plant, but when you do embryo or apical meristem, you know it's virus-free.
Oh,
Okay.
wow,
Because-
that's kind of a key point. I
I'm-
didn't get that.
That's why you that's why you ideally do that. For general propagation, when you just do it, you do not have to. That's why we did tobacco leaf. But for when you are starting a breeding program, you're starting something and you find a really cool plant, it has a weird discoloration. You think it's variegated. You're gonna isolate the apical meristem, start it, and then it doesn't show the weird variegation, and you are scratching your head like, what's going on over here? It could be caused by a virus. It looks really cool, but it's not actual variegation. It's not changes to its genomic makeup. It's just a virus that's causing those symptoms.
Right.
And you don't really wanna.
So can you, so the plant could be virus, but if you take one of those two places,
Yeah.
it will be a virus-free clone, but otherwise it wouldn't necessarily.
Yes, because if you have a virus or something, you do not know that why is the leaf showing that symptom. We know that our plants that you guys prepared, we didn't actually collect those from nature, or like it wasn't growing in the greenhouse. We've been growing those in one of those plates
So they're
for so
virus-free.
long. We know those are virus-free, yes. Exactly. So basically, I really hate when they say sterile, because it's technically not sterile. It's aseptic. If you have something living in there like a plant, it can be sterile because that's organic, that's living. But for this, basically, we just gonna remember this part. In vitro culture of plant parts, quantify nutrient media, controlled environmental conditions. So basically what you wanna remember what plant cell or tissue culture is. Basically it's a aseptic condition. So you have no pathogens, no fungal, no bacterial, no viruses. You wanna make sure that you mention that it's a specific nutrient makeup. You control everything. In greenhouse, you control more. In a growth chamber, you control even more. In tissue culture, you control everything.
So in vitro is just basically micropropagation.
It's Latin for in glass.
Okay.
That's why I like to use aseptic, not sterile, because technically it is not sterile, because then you would have no living thing in there. But some people like to use it interchangeably. In vitro sterile, basically. That's why these have no contamination. Once I open it, I exposed it, yeah. But not before we actually have it. Okay, so this is nutrient media. So I think we're gonna talk about this is a really good picture of contamination. like it just kills everything. It uses the nutrients that you put in there for the plant, and fungal spores, bacteria is a lot better at uptaking those than your plants, and it will actually kill your plants. It will overgrow and kill your plant. The tissue culture, it's basically not just you can't just do research with it, but you can also do a lot of commercial. Like when you find a new cultivar and you wanna propagate multiple of them, so you can get that cultivar out to the market.
Is tissue culture the same as what we were talking about?
Yes,
Okay, so
tissue culture is basically
just wondering if there's an
inverto.
actually defined difference in the definition.
So basically, it's an inverto culture. That's when you are working with plant tissues.
Like the tobacco that.
Yeah.
So leaf
That's
cuttings,
tissue.
leaf cuttings is tissue. Mary's
Because you
it's
can
still
do-
tissue.
Yeah, because for basically, you see, like it's plant cell and tissue culture. That's the same. In verticulture could be different because you can do bacteria. You can grow bacterial. You can grow E. coli for like production of insulin. So when we say tissue culture, we actually specifically
Plants.
plants. Yes. When you say in virtual culture, you can do a lot more different things. Any other questions? Really good questions. And again, you control everything with this. You control light, temperature, humidity. Okay. And the other thing, it's really important for breeding programs. When you want to get haploid, double haploid plants, you want to do embryo rescue, like I said. Those are really important. And basically, again, you just want to create large number of uniform copies of plants, like I said. When you want to build up a population of it from a single plant or a single plant cells. And then you have the morphogenesis, basically how you can go from one cell to a whole plant.
Is this showing, this picture right here is showing how you'll get from one cell to the callus?
Yes, this
to
would
eventually
be,
shoot growth?
yes, this would be one cell with no cell wall. So you can actually dissolve the cell wall. We're gonna talk about that a little bit later. But yes,
So you said that to get your callus, you have to have equal parts of auxin and cytokinin.
it depends for, in general,
Okay,
but there is always an exception. We have specific makeups for different plant species.
when do you, or how do you manipulate your callus to either increase or decrease auxin or cytokinin to get the shoots or the?
You move it to a different media. You just make a different media and you move it, you move the plant. Once you put it in there, you can't change it, but you can always really easily just grab the plant, move it to a different media, and then the callus is gonna start actually growing either roots or shoots, depending on what you want to do. Really good question. Yes. So again, this is the apical meristem. This is the only virus-free part of the plant. If you have some kind of contamination, this is how you can save real specific cultivars. And this plant shows you don't necessarily have to dissolve the cell wall of the plant, but we're gonna talk about why you would do that. And you can see like these callus, you put it in a different media, and then it's gonna kind of have this green coloration. Then you're gonna have like growth, and then 4, it's like basically completely different. So 1 is callus, 2 is when you start getting a color, 3 when you start to have like a shoot structure, green, it looks like something, and 4 is when like you have the roots established and it's green. Virus-free plants and germplasm preservation, basically, instead of having to have like, I don't know how many corn plants to save, the line, I can have one plate and that has like how many plants? Like, I can regenerate like multiple plants and I can stack those, keep them in the dark somewhere, and I have to touch it every 3 months when I'm gonna put it on a new media to keep it alive.
So it's just like storage.
Yeah. a really efficient storage. Again, yeah, these pictures show really well like what it looks like. You know, like you can do it. That's not a closed chamber, but if you are in more like you guys saw in our lab, it's a lot more condensed. We don't have as much space. It's a university. It's more research focused, so we have to be a little bit more efficient with things like that. And haploid and double haploid plants is basically how you're gonna obtain homozygous plants for breeding programs. And then you can do a bunch of cool stuff.
So do you do actually the splitting there in the tray? Do you take like a cell from there, take the meristem from each, and then like kind of like zygote it itself, and then culture
So
that?
you have to get like the plant in a real specific stage. I'll be honest, I'm not 100% sure how to do haploid and double haploid. I know it's not that complicated. I'm not that
You just
familiar
have to-
with the process. If you want, I can look it up, the process, and
So, I was just kind of curious.
Yeah. because I can look it up for you and send it to you email, or I can make an announcement. If it's like, I know they do it at the wheat and research breeding building right next door. And there is that company that specializes in that, and they make a butt ton of money off of that. So this is the embryo rescue when you do a hybrid, and you see that's the embryo, like the actual storage is not functioning, like the pollination occurs, but the embryo is not viable by itself. And just like what I did with my, like it looks something like this, and before it gets mature, you can get the embryo out, put it on, just like I did basically with the corn, except the corn was viable, but I wanted to get the embryo out to reproduce the line quickly. They can do that exactly the same and save the really weird hybrid or cross, whatever you are trying to do.
So basically, the hybrid would have been sterile otherwise, but then you
Yes.
go in and actually.
So it's not technically sterile. It's
Ish,
just
so. It
It's
wouldn't have
just
been
the
viable.
genes are not completely not completely compatible. So it wouldn't produce an offspring that can survive.
Right.
But if you get, so you get the embryo, but the seed is not forming correctly.
Right.
And by doing that, you can rescue the embryo and save the plant, which most likely we will be able to reproduce.
Oh, the next one. after
Yeah,
that.
that can actually reproduce in most cases. Not all cases, but it usually can. And like we talked about the tomato hybrid, where you had the really big virus coming through, and then you found that, or they found that really, really far species, or not species, it's the same species, but it's a really far variation, cousin, yeah, whatever you want. It's the same family, but not the same species. So you couldn't interbreed, or they couldn't interbreed, but by doing embryo rescue, they could cross those, save the embryos, and that from the progeny that you got from those were actually resistant to that tomato virus. And then by breeding that new progeny, again, the new tomatoes, you could pass down the resistance. So it was able to breed, the offspring was able to breed with other tomatoes to get the resistance through, but it just needed that little help with embryo rescue.
So what disease did it become resistant to that you talked
It's
about?
a tomato virus.
Okay.
I'm not sure.
Mosaic, the tomato mosaic virus?
That doesn't kill it. It's not the tobacco mosaic virus, but it was like, I can't remember the exact virus, but the tobacco tomato mosaic virus, that can attack it, but it won't kill it. It's just, this virus would actually kill it. And that's why they had to do that. And then cell selection, basically, if you wanna do some, if you do some plant engineering, breeding, you can actually make a selective media that will only let plants with a really specific properties to survive, so you can collect it. Basically, if you do a random mutagenesis, or whatever, and you wanna get heat stress. You can do a heat stress maybe or something you want. You can raise the temperature, and even if one cell got mutated and has the right mutation to survive the heat, from that single cell, you can regenerate the plant.
So basically, you're doing evolution in a very quick sense.
Yes, or you can do genome editing where you just introduce the gene and engineer it. So when I did my tomato experiment, I had thousands of tomato plants that I used a chemical mutagen on it. And I just wanted to get the red color. That was the only thing I wanted to get, red color. I didn't care about anything. And I had 4,000 plants. We also compared it to CRISPR, where you just had like 4 petri dishes. We genome edited them, put them in there. We had a selective, we made them resistant to herbicide. And if they became herbicide resistant, we had the gene, basically the red color gene from purple, or we knocked out the purple gene. So it would be red. And if that survived, then you could actually just plant it and you had red plants, red fruit producing plants. And it basically tells the same thing. And this is the same thing with protoplast fusion, where you dissolve the cell wall and you can introduce the two cells to each other, they combine genetic material. That's how they
become like, what do you call it, karyotic
then?
Like multiple nucleuses in that one cell?
So basically they were combined the genetic material. So like this, you wouldn't be, you have the properties of 2.
Yeah.
But the genetic material combines.
So you would get the 2 nuclei together, basically.
Yes, it would merge. Yeah. So yeah, basically, and that's when they were doing the tomato with potatoes, but they didn't really work out when they first started. And now they are getting closer, but I don't know much about that. And then production, secondary metabolites.
So how does it make the secondary metabolites?
It's basically you provide a plant or bacteria with the nutrients, and you can actually just, it's gonna produce it. You can.
So like if you want salicylic acid. For
Yeah.
example, you could
So
do that.
insulin, the way they are not getting the insulin from pigs anymore, what they do now is they actually have E. coli engineered to create insulin. So they just grow insulin, they grow E. coli in huge tanks, like in virtual conditions, and then they just, once they are done, they press the E. coli and then remove the insulin.
So these are all the reasons why micropropagation is something we do.
Or you can do like, the enzyme production, which I'll be honest, I'm not really familiar with like the production side, but you can, as you create, like I said, as you create the media, you can put different amounts of growth regulators, which can encourage secondary metabolite production. And since you're controlling the heat, cold, light, all of the conditions, when you stress it, it's more likely to. And I'm sure they have like a really specific protocol how to achieve it, which is probably it's probably a trade secret, which not really like I wouldn't think that it's just online. We don't want much competition. And like I said, we can talk about plant genetic engineering, which is, you guys can see the colors. What I'm going to do is I have different lines of callus, or line mean like different gene, corn with different genes, and I can just put these calluses in high temperature and see if they're gonna survive the high temperature, and I can evaluate if that gene is actually helping the plant to survive in higher temperatures than the regular wild type that doesn't have the gene.
So that's what you're doing with your corn and your really.
Yes. So basically, instead of having to wait for the greenhouse and wait months to grow, I can just do this in 2 months or less than 2 months, actually have results. So it saves time. You can do a lot of like heat stress tolerance, cold, drought, salt. Like I said, you control the media, you control everything. You can put toxic metals, you can put salt in it, you can figure out which plant is resistant to virus if you inoculate it with the virus. You can do a lot of cool stuff. Toxic heavy metals, basically you can engineer plants that can take up heavy metals. You're gonna collect those plants, you process it because they're gonna store the toxic minerals in their nuclei. When you harvest that, you cleaned up the toxic spill.
So what do you do with the plants after that? After they've taken, I mean, because you use that to like remediate your soil, so to speak.
You
right?
have to
What do you do with
store
the plants
it.
when you're done?
You have to store it. You have to dispose of it correctly. I'm not sure. It depends on the material what you are cleaning up. They might burn it at high temperature. Don't quote me on that. Or they might just like seal it up, bury it somewhere where it's like
Somewhere else.
Yeah. Depending on, as long as it's not out in public, when anyone can just walk out to it. And you can actually recreate something like this. And yeah. Basically, petunias, you can, actually there was a case of petunias where they created orange petunias. And it got out to the wild in Europe and they were not really happy about that. Because Europe is really strict about genetically modified organisms. And yeah, and like I said, you can actually engineer plants, you know, like this is petunia, and you can have like toxic metals for cleanup. You actually create toxic metal resistant plant for cleanup.
When you're testing that, do you just put some of the
Toxic.
toxic in the media and
Yes.
see what survives? Okay.
Yes, basically that's it. It's really simple. Okay, so basically the key points, the definition of tissue culture, which is where you control the media, you control the temperature, you control everything. And that's basically the biggest takeaway. And if you go back again, I can get that for you guys.
How long, you said you can store these calluses
Indefinitely, indefinitely.
What?
They
How
don't.
does, the media is just that saturated with nutrients?
So
that
you
it
can, you have to actually move the colors every 3 weeks or every 3 months to a new media, but you can store the colors indefinitely. You need to replace the media,
Okay.
and it is room 25 something. I don't know.
Yes, 27.
Yeah. So
Yeah.
so this is what I want you to guys to take away. This is like the biggest sterile in verticulture of plant parts on defined nutrient media, controlled environment.
On the test, it will be sterile, but you're saying aseptic is.
It's
what it
I
is.
prefer aseptic. That's more accurate.
But what will be on the test?
I-
This is the test, I'm gonna yell at you.
I don't know. I'm not
Well, just
writing
remember,
the test.
aseptic and sterile go hand in hand, they don't have the same thing, but people.
Yes.
use them interchangeably.
It's like they use synonym. They use it as synonyms when they are not synonyms. Yes.
It's like a tissue and tissue, I mean.
Okay. Let's get to the next one, which is we're gonna talk little bit about the production and what we're doing. And micropropagation or clonal propagation. Again, we use those on. Use these two words interchangeably.
Which two words?
Yeah.
The micropropagation,
Micro-
clonal
okay.
propagation. So basically, it's a propagation method used in Verto, which says within the glass, just because when they were actually doing these, they use glass and they are reproducing living organisms in a controlled environment. It could be a test tube, petri dish, or a magenta box.
Magenta, why magenta?
This is what it's called. I think it used to be magenta color, but
It's not magenta.
no, I'm assuming it's a trademark name and they just stuck with it. So the definition, it's an asexual reproduction resulting in genetically identical copies of a cultivar. Basically like we thought. genetically identical copies. No, it's okay. Now it's fine.
I can see why I should ask.
Now it's fine. If you opened it, that's fine. Don't worry about it. You can touch it.
That's what I wanted.
Now it's all messed up.
Immediately after I did that, I was
Is the media,
I actually brought things that you can touch specifically, but
Especially those.
it's okay. It's okay. Nothing.
Bad.
You can actually convert these from with the tobacco media. That's okay. I should have specified. That's my.
No, it seemed very.
is
obvious
this
in
gonna be,
hindsight.
you know how people have you touch something like this and then they grow bacteria and tell you how dirty your hands are?
Yeah.
Is this what this is kind of?
Yes, kind of like, but it's like
It's made different, the media
Yeah.
made different.
it is specific. That is the magenta box. It has a specific media for
Yeah, when I
carnivorous
touched
plants.
It got like.
For what
very
kind of
liquidy.
plants?
And that is for carnivorous plants. That is for tobacco specifically. Okay.
Why did it get liquidy?
Or
after
maybe
I
just,
touched
it's
it?
just.
Because of the pH, it's a lot softer. The type of media, actually, that is made from tissue culture, but that's made from gerlite, not tissue culture.
This one's agar, that one's.
Yeah, so you guys can actually feel the difference. That's fine. This is why are we here, so you guys can learn. Okay, basically the colonial propagation is we're gonna create a very large number. Okay, guys, let's bring it back.
Yeah,
We don't have much time. I don't wanna actually
yes. we are
hold up
focused
you guys,
on-
but basically the colonial propagation is when you, it basically says that you create a large number of plants. There we have some, there you go. I'm gonna help you out with there.
Give them to me now, she doesn't need them.
Here. Okay, basically we're just gonna create a large number of copies of something in a short period of time, and it's aseptic. This is the term I prefer instead of sterile, aseptic.
Okay. Aseptic.
If you say aseptic, people gonna be like, you know your shit. Basically, they're gonna be like,
That was.
they know their stuff. Yeah. And basically, again, everything is controlled. And yeah, ornamental, you can use this for ornamental, food production, research. They have many. If you can imagine it, they use it for it. Some people actually sell. I had Liv ask me if she could do, she brought her own container. She wants to do carnivorous terrarium, tissue culture terrarium. So she's working on that side project.
That's cool.
So yeah, a lot of different breeding, food production.
You said you need forest and plantation.
Basically, when you don't have the right climate, it's really hard to grow a tree or propagate it. Guess what? You can create your mini trees, not taking up a lot of space in a lab, and then just plant it outside, transition it.
That's cool.
So you don't necessarily have to have the right climate. Not everyone can have a huge greenhouse to get everything. And it's a lot cheaper, in my opinion. But that's a little bit different. So basically isolating shoot tip, lateral buds, that's because we know that that is virus free, but you can also get from leaves, embryos, other things like that. But the apical meristem, axillary buds, those are 99.9% sure. Those are virus free. Okay. You're gonna use your plant regulators to control what you want to do. This is where my thing gets a little bit out of. And this is the magenta box. You take a cutting off of it, but you sterilize from the sample, you can grow it, and then you can just put it outside and you're gonna have a bunch of plants. You can do that with your tobacco plants. So I actually still have mine from 2 years ago, this big, huge leaves. I'm really tempted to actually roll some
You
cigars.
just have that in your house?
Yeah.
Really?
Yeah. Yeah, just roll some cigars. This is basically petunia. It's the same method that you used with the tobacco, you just cut squares with the couple and then put it in media, and you get a bunch of new petunia. And there are many. We have direct organogenesis and somatic embryos. Basically,
So explain that definition, that's what I was trying to ask.
when you actually create plants without the formation of callus.
Okay,
So you do not have callus in here. You grow from like this plant, a new plant, no callus. And like I said, please write down your questions on the note cards or just ask me. This is why are we here? I want to make sure you guys are feeling ready for this upcoming exam.
Is it a different process to do an organogenesis, or is it different genuses?
It's different. It depends on the plant.
On the
For some plant, you don't need callus. You can just create like that. For some plant, you have that step of getting the callus. Just a heads up, I can guarantee that you will need to identify callus on the lab practical. That's gonna be, you wanna make sure you know how it looks. But you can see that there is no callus, no white, undifferentiated cells, like a bunch of them come together. It just goes straight from plant to plant. There is no callus. Direct organogenesis, somatic embryos.
Are throughout like genuses, are calluses always gonna be generally white, or do you get like a darker color?
It's generally white.
Okay.
But you can have a little bit of discoloration. I think Kendra is outside. I can just. Okay. Did she leave? I think she might have went into the restroom, but maybe, we don't know. Okay. So basically, somatic embryogenesis, we can generate plant embryos that are not generated from embryo cells. And so somatic embryos are formed without fertilization, and they are genetically identical to the parent matter. Like I said, you can basically generate new plants without callus and without, from a plant part that is not generally involved with sexual reproduction.
So that means like, you know how you were saying the immature embryo, this process does not involve like the embryo,
No,
any other plant, any other
Basically,
part.
as you can see right here,
It's like
it just grows, the embryo grows from like a leaf, like that.
Okay.
You might see this with succulents. When you actually break down a leaf and just let it sit down, it's gonna push out a brand new plant from the leaf base or where you broke it off, without any callus. That would be an example of the somatic embryo or direct organogenesis, without, of course, using tissue culture, because you don't necessarily have to do it in vertical condition. Yeah. We went through this. So what is a callus? It is a wound response. It's an unorganized mass of undifferentiated cells. And it is a response to protect the plant. As long as you mention it's a wound response, unorganized, and a mass of undifferentiated plant cells. Or you can say proliferative. But as long as you just remember wound response, unorganized mass of differentiated cells. It does not have a specific function. It's just like the apical meristers. So,
Come on, keep going. I'll ask, you're almost getting there. Yeah, right here.
Okay. So the sequence of embryo formation.
I'm kind of curious about the shapes in particular, because that.
yes, I was confused about the shapes.
Okay, so basically, you have the explant. This is what you use, right? And then you can have the accent. This is a plant cell. So is this confusing that this is like
What is it?
So
I'm not sure what we're.
That is a plant cell with no cell.
Oh,
Usually plant cells are not circular. Otherwise, you wouldn't not circular or not spheres technically, but we just only see them in 2 dimensions. Right? It would be a really bad building block of a plant if like that's why they have that square shape. So you can build a structure from it. If they were circular, plants would just look like garbage, right?
So you isolate that single cell from the eggplant?
Yeah, or you don't necessarily even have to isolate.
It's just
Single
saying
plant.
that
Yeah,
the
it
cells
just
are in there.
the cells, and then the cells start to replicate, and then they're gonna all of these cells create the callus. That it's replicating, it creates the callus, and then you get the callus fragmentation, clumping, and you can have the globular, the heart shape, and torpedo shape.
Does it change shape or does it just actually, like, does it start globular and go heart shaped, then torpedo shape, or is it just one or the other and then it goes to plant?
It depends on the species. So you can have one that actually changes, but you can just have like one shape, like heart shape. But it's generally, this is how it goes, the shape changing. From like, you see how you get, and then you get the plant. As the cell starts to differentiate, right? Like you actually have the clumping of the callus. So you're gonna have a globular and then the heart shape, and you can start seeing like, okay, this is gonna be the shoot system, this is gonna be the root system. You're gonna have the torpedo shape when it's like even more, and then these gonna actually differentiate into leaf.
Those are real cotyledons.
So these are not these are real cotyledons.
Yeah,
These are not.
Yeah, not those, but when you're done, when it's gone through this
Yeah.
whole, it goes through. Okay.
And then this is just, I'm not sure if you guys can see, like basically the way we number them. Callus would be a number 1. When you start seeing, this is not really sharp enough, but when you start seeing kind of like these shapes, you can actually see when the callus start to have like these clumps on it. Like you're gonna start to have, that's we call number 2. When like, yes. And then they start to have like a basic root system or shoot system, that's number 3. And when you have the shoot system and root system, that's stage number 4. But we're gonna talk about that a little bit later. But this is what you can see, like callus, and it grows like this. And I know we have a really good picture. I'm gonna skip these. Like callus.
Ooh, what's picture number C?
I'll be honest, I have no clue. It's not callus. I think this is probably plant parts in like tissue culture media, and they like drop it like you see, like how it is like that. I would think that's tissue culture media.
Okay.
Since you're just reading, you see this is looks like tissue culture media that actually creates itself.
Cool.
Okay. See, and then yes, and then these are the 4 stages of micropropagation. The initiation of the aseptic culture, shoot multiplication, rooting, and transplant into soil. Stage 1, this is where you get the aseptic shoot. Stage 3, or callus, and starting the formation. Stage 3, or stage 2. Stage 3 is when you start to have the plant actually getting ready into a magenta box. Stage 4, tomatoes. And this is like one of the best things from like few of these leaves. Like if you have a tomato that you really want to save, and it's like dying because you did research and you messed up and you need to get more plants off of it, you can actually go through and regenerate the plant without actually, messing it up too bad. I haven't had to do that yet.
yeah,
but
sounds like you might.
I got close a few times, but not there yet. Okay. So basically what you, like we talked, what you have to consider for tissue culture or micropropagation media has to be really specific. Although we have like a general mix that you can use without any like issues, you can get really specific medias for specific plants for the best performance, the fastest, everything. Environmental, you don't want to cook your plants, you don't want to freeze them. You want to make sure you provide enough light. And browning of callus can usually occur due to oxidation, due to phenolic compounds.
Can you explain that just a little bit more?
Yeah.
Basically,
Like what about stage 1 and browning?
stage 1,
What is the browning in stage 1?
browning in stage. Sir, can
Like
you?
the callus just
Oh,
gets decolored or what? Do you-
okay. I think we have. Look at the. Like, you can see some of the browning on there. Basically, as it uses up, like over here, as the callus uses up the nutrients from the media, it can create compounds that, since it's a closed system, they can get toxic. That's why you need to move the callus every 2 to 3 months.
Okay.
Browning isn't good. Browning's not.
No, browning is not good. You want to avoid that. And there are some benefits of the micropropagation. And I think we're gonna have a short break after this, so you guys can ask questions. And yeah, so basically you can propagate a large number of plants real fast. It's a lot faster. Like I said, I can reproduce hundreds of plants in 2 months instead of spending 2 weeks, or instead of spending 2 years to get the same amount of plants. It's pathogen free. You are not actually limited by season. I had to grow corn in the greenhouse this winter. It was not fun. Let me tell you that. It is not fun. You don't want to grow corn in the greenhouses. And sometimes you need to. This is a lot easier. Long-term storage, like I said, every 3 months you have to move it to a new media, but otherwise you are not really limited by space or anything. And breeding programs, double haploid, maintaining heterozygous lines, a lot easier. You don't have to worry about cross-contamination, pollinate, anything like that. So conventional propagation benefits, minimal equipment cost. I would argue with that depending on what you are doing and how big you are. You don't need a lot of experience, inexpensive. It's a little bit of a trade-off because you need to spend more space. You know, heating a greenhouse when it's really cold outside can get really expensive. And, you know, you can get like growth chambers or something like that, high tunnels for grafting, thing like that. Disadvantages for micropropagation: you need to be trained and you need specialized equipment. Kind of. Instead of an autoclave, you can use a pressure cooker. I mean, it's gonna slow you down, but if you're not doing a lot, just small amounts, that's gonna be fine. You need a lot more expertise. You wanna avoid contamination, right? You need protocol for each species, and people who have specific protocols, they might not be willing to share it just because they spend a lot of money on it. If you look up some literature, you might be able to find some, but not for like large production. Labor, it's some more expensive since you need to have someone who's actually what they are doing, or just hire college kids.
Yes, do they usually want people with degrees to do this, or do they get?
So for research, yes, propagation, I would say probably you need to pay a little bit more than you would ideal, like, you know, just with someone who's like, but I don't think there is like that much of a labor cost difference, especially Like it says 50 to 85, but when you pay 15 instead of 10 to a college kid or something, you know, like, especially you can get a lot more. Stage 2 is repeated cutting, transferring. So you wanna make sure that you keep everything, rooting, transplanting into soil. Like the last one is probably the least expensive. Yeah. Because you don't need to be specific. You don't need specific training to do that. But stage 2 is like you wanna have the callus transferred when it starts to produce some shoot system. Stage 3 is when rooting. Remember, stage 1, callus. Stage 2, you start to have the weird shapes, the globular, the heart shape, torpedo shape. The callus starts to have those clumps on it. Stage 2 is like that stage. You kind of start to have the shoot system. Stage 4, you have, or stage 3, you have rooting. And stage 4 is when you have the plant in the magenta box or wherever, and you can just pluck it outside.
How hard and how expensive is it to get the ingredients?
You can order it on Amazon for 20 bucks. It is not expensive at all. Or you can just make your stock solutions a lot cheaper. It's gonna be overall more expensive, but you can make a lot more. And again, a lot of people use just regular sucrose. We use specifically like research grade, so we have to get the high purity stuff. For production purposes, you're not gonna get the most expensive, like this clean sucrose. You're not gonna get the best agar. You can get gelatin, like, you know, you can see that a lot of like productions, it's a lot lower grade material because they just like, it's,
Like you're propagating for, let's say, like purposes versus research purposes.
yes, there is a huge difference because we need to produce repeatable data, and it has to be a lot more controlled than if you are just doing something for propagation purposes. And if one batch maybe does not perform the best for whatever reason, you can be like, as long as it's not dead, you can use it. Right? Like if you guys work at a nursery or something, and some plants are a little bit shorter, grow a little bit, you're like, oh well, you know.
How often do nurseries do this?
So I only know a few places that actually had their own program. Kagawa greenhouses did their own. I know, but they stopped doing it just because of their cost. They didn't really see the benefit of it, but you know, you could technically, and it's like not season dependent. The costs or concerns with micropropagation, contamination, you can also really easily mislabel stuff if you're not careful. From callus, I can tell if that's a corn callus or tobacco callus. You're only gonna see it once you actually start to get the shoot system and overproduction. So if you produce way too much, you're gonna drop the price real quick and it's not gonna be viable. So you wanna make sure that it's economical. You don't wanna push out plants 24/7. And high cost, relatively high cost setting up a lab. So depending, like I said, you can get fairly, like I've seen laminal flow hoods online, $500 used. Not that bad.
You said you could use it instead of.
Pressure cooker, same thing, high pressure, high heat. You don't necessarily need the autoclave. Growth chamber, you don't really need it. We need it for research purposes, production purposes, as long as you can keep like we actually keep at 26 degrees Celsius, which is about 75-ish. So as long as you can keep the temperature 75 degrees and give it light, it's
It's
gonna.
included in its germ chamber then?
Yeah, you can. Yeah.
Basically just like a.
Yeah, as long as you can control the temperature, you wanna be careful not to cook it because it's a greenhouse in a greenhouse. That's the only thing. So you don't wanna leave it in the greenhouse.
What about, I forgot the tool that you use to constantly stir while you are
The stirring rod.
Is it just a stirring thing? It doesn't
Stirring,
do,
no.
it doesn't
That was
like
the
heat up or
magnet on it?
anything?
Yeah.
It has a magnet in it.
It just stirs.
It is. You can buy a heating plate and the stirring rod on used for $50. Like these are not that expensive, especially if you are getting production grade, not research grade. The prices drop dramatically.
Yeah, and then you guys distill twice, which he said it's literally for research, you don't have to
Yeah,
distill.
exactly. You can get the regular jug of water from, the DI water
Makes
from.
sense.
So basically, this is just tells you the number of micropropagation in the US. I'm not gonna go in there. And blueberry production and production of bioweeds, like what kind of cool, a few cool stuff like propagation or definition of micro. So propagation is creating a large number of uniform copies in a short period of time in a completely controlled environment. Okay, basically that is it. I'm gonna let you guys have a 5-minute break. I think that is a completely reasonable thing to do, right? I think.
Yes.
I have a question. So you said, hey, I don't want to take over.
Yes.